to identify a naked polyhedral, a naked helical, an enveloped polyhedral, an enveloped helical, that you know what capsids look like, you know what spike proteins look like, you know what an envelope looks like, you know what an envelope is made out of. And the only ones that you're expected to know RNA or DNA virus and what the genome is inside of the virus are the ones that are going to be on our note cards. So any question that I ask you about anything that goes against the central dogma, or even if it goes with the central dogma, are how do they hijack our proteins, our enzymes, and what enzymes of ours do they use versus what enzymes of their own are they going to need. So what we can do also is, I was going to say ask Dr. Welsh, but I'm going to also make a video for us tomorrow, and I'll go do some review tomorrow I'll do like a short video, hopefully short. So, but try and just work through it yourself. If it doesn't go with the central dogma, then it has to have its own methodology of reproducing, transcribing, translating. Okay. So we stopped last time talking about zoonotic diseases. Oh, you know what? I'm going to jump around a little bit. Forgive me for jumping around. But I'm going to start here today under viral control, and we're going to watch this really quick. So this is the NPR one, part 3.
Gods, misaligned planets, stinky and plenty of bad ideas. For most of human history, we had a lot of bad ideas about how we were getting sick. Angry gods, misaligned planets, stinky fumes, and plenty of bad ideas about how to prevent it, like bloodletting, large holes in the head, arsenic. Things started changing only about 200 years ago. An English doctor named Edward Jenner decided to take a closer look at a promising folk remedy. See, milkmaids, they'd get these blisters on their hands, cowpox blisters. People said that if a milkmaid got cowpox, she would never get its deadly cousin smallpox. So our doctor decided to put this milkmaid wisdom to the test. His first step, he wiped gunk from cowpox blisters into a cut he made in the arm of his gardener's 8-year-old son, James Phipps. Sorry, James. James got better pretty quickly because cowpox is usually mild in humans. Then, the doctor made his bold and risky move. He injected James with the similar but deadly smallpox. James didn't get sick. The milkmaid cure had worked. And with that, Jenner invented vaccination. Here's how it works. When you inject weaker germs into our bodies, our immune system takes care of the problem and stays on the lookout for similar looking germs. So when the really bad guys come, we're ready for them. This was our first safe and effective way to fight disease. About time. After that, things started happening fast. Over the next 150 years, researchers from all over the world joined this fight against germs, discovering them, developing vaccines, killing bacteria with antiseptics and antibiotics. So what did that do for us? Consider that in 1900, the average person lived only about 30 years. Today, most of us live to 70. We were so successful in such a short time. In 1967, the US Surgeon General reportedly exclaimed that we had closed the book on infectious disease. But the truth is more complicated. See, what happened way back in episodes 1 and 2, when humans were cutting down forests and catching new germs from wild animals? That's still happening, but now it's on a scale our ancestors never could have imagined. We've cleared about half of our planet's arable land, and we're still cutting. Remember, these wild places are where undiscovered germs live. By getting rid of their homes, we're forcing them to find new ones near us. And there are more of us than ever living closely with animals. Throw in some planes, and the next thing you know, a new germ can turn into a worldwide pandemic practically overnight. Think SARS, HIV, Ebola, bird flu, Zika. These are just some of the viruses plaguing us right now. And we haven't even mentioned antibiotic resistance. So the story continues. Some scientists say the next chapter is us looking for ways we can live together with germs. Germs in their homes and us in ours. Let's just hope our big brains keep working overtime so we find a way to stay one step ahead of that nasty bunch of fast-changing, human-killing microbes, the germs.
So obviously this was quite a few years ago, right?
For most of human history,
Okay. So I'm gonna play you one more.
I had a little bird, its name was Enza. I opened the window and influenza.
Perhaps until recently, Spanish flu has been what some people call a forgotten pandemic. But it was a huge global calamity. In terms of number of deaths, we're saying 50 to 100 million, far greater than the casualties of the First World War. Roughly around April, May 1918, when the carnage of the First World War is graphic and horrendous. But at the same time, this unknown enemy is starting to sweep through the US, Europe, the trenches, and it is recorded by the Spanish press, and it becomes dubbed as the Spanish flu, because they are neutral. Well, at the time in the 1920s, they estimated that maybe 20 million had died. By the late 20th century, that figure had been up to 25 million. In the 21st century, with scientists and historians now getting together to work much more closely, that figure has now been upped to 50 to 100 million people killed. This virus killed more people in the first 25 weeks than HIV/AIDS has killed in 25 years. Horrendous. I mean, those mortality statistics are staggering.
We don't know exactly why some flu strains are much more severe than others and why some of them would be more likely to kill people or cause very severe disease.
What is particular about avian and pandemic viruses is that they replicate deep inside our lungs. When our cells detect this and trigger a very strong innate immune response, this leads to an influx of white blood cells and fluids into our lungs, and it restricts the amount of airspace that we have to breathe.
The symptoms are very graphic and very, very striking. So, it's said that some people drowned in their own phlegm. So if it was hitting or affecting the lungs, people were spurting blood from their ears and their noses. Descriptions of people turning blue or purple. So, it was really, really severe, and people just dropping down in the streets. Those that succumbed to the flu were in the age group 20 to 40 years, which is very unusual for an influenza epidemic or pandemic. And scientists and historians are now still looking to see why.
There's really two ways that people died in 1918. They either died from the initial flu infection, or they died from an enormously strong immune response to that virus, where their lungs just filled up with fluids from trying to attack the virus that got into their lungs.
So traditionally, everyone sort of said, oh, well, it's the trenches, and, the conditions of the war, and that's why it's so fatal. But when you think, well, how did it spread so rapidly around the world to places that are not part of the Theatre of War? I think the traditional explanation is it starts in Kansas in an army camp, and then the troops bring it over to the trenches, Europe. But there's still many, many unknowns.
So mathematical modellers have long been interested in the 1918 pandemic, and one of the questions is to try and disentangle how it spread around the world and exactly what the routes have been. Given perfect data, you could work backwards and try to infer where it's come from. Whereabouts does it arrive in a country? Does it arrive multiple times? Or thinking about multiple countries, where is the first place? If you understand the origin of a pandemic, then you understand something about where it's come from, and maybe you can stop that in future.
Almost certainly it was a bird flu originally, but then we also know that that flu, or something very similar to it, was in pigs at the same time. Maybe it's somebody who works very closely with birds, or who works very closely with pigs, or has some other exposure like that. One of the big, really open questions in flu research and flu public health is, how does the virus adapt from one species to another? How does, for example, a virus in pigs or in birds get into humans and then start transmitting? Then the key question is, how does the virus then adapt within that person to stop being a pig virus or a bird virus and become a human virus? In other words, to be able to transmit from human to human. And it's probably some very quick evolutionary process that is going on within that human, and in many cases won't be successful, but in a few cases will, and that's when the virus can start spreading between humans and become a flu pandemic and then stay in humans as regular seasonal flu.
Just as people were celebrating Armistice Day, there was hope that it was dying down, but then it resurged and a far more deadly and virulent strain emerged. So this second wave really spread like wildfire, affecting a third or a quarter of the population of the world. And it was short-lived, so it petered out not long afterwards, maybe 1920, 1921, but it was in a sense short-lived.
What happened is that virus continued to transmit among humans. In fact, it continued until 1957 to transmit between humans. And probably what happened there is that the virus adapted to humans. It wasn't this avian or swine virus that came in and was accidentally more pathogenic in humans. In our lab here in Cambridge, we studied both seasonal flu and pandemic flu from the standpoint of how does the virus evolve? And how can we predict that evolution and make vaccines to try to artificially evolve the virus both in the laboratory and in the computer to understand what it's going to do next and to make vaccines against those strains that will evolve in the future. So this is the basic science of studying evolution and trying to predict that evolution, something that nobody's ever been able to do for a complex organism. And then working to use that understanding to make better vaccines. On the pandemic flu side, we do a similar thing where we look to see how that virus is changing, that virus, how it changes in birds and how it changes how that virus changes in other species, and to try to see what possible variants out there might be. And again, to make vaccines that could protect against all of those variants that we see.
Flu is very difficult to eradicate because it's constantly mutating and changing its genetic information. We work on the influenza virus, and in particular on the enzyme that copies the genetic information of the virus when it has entered our cells. Specifically, we're interested in how this enzyme can do this activity and how it inserts mutations in the viral genome that leads to the formation of new strains to which we need to develop new vaccines. And we are also interested in when this enzyme messes up, how it triggers the innate immune system and helps cause disease.
We really don't know why we haven't seen a pandemic as deadly as the 1918 pandemic. Scientists and public health officials are extremely concerned. The National Risk Register, which is the way that our government plans what are all the risks to civil society in the UK, there's only 2 events that are up there in terms of the most catastrophic, and that's a terrorist event or a influenza pandemic. There is a real threat that there can be such pandemics. For example, the H5 and the H7 flu viruses that have infected now close to 2,000 people and killed about half of them. There has been with the H7 viruses some transmission from human to human, not very efficient, and it hasn't really taken off. If either of those viruses did adapt to really transmit well between humans, there's a real concern that it could be as bad as the 1918 virus. And this is why there is so much research done for the public good to try to understand what's happening, and so much work done by people in public health and preparing in hospitals in case something like that happens.
Well, experts like to say it's not if, but when. Coming as a mathematician, it's happened a lot of times in the past, it will probably happen again. The 1918 pandemic is difficult because of lack of data, but the most recent influenza pandemic we had was in 2009. And from that pandemic, there is some very good data. If we understand how the 2009 pandemic spread, maybe that tells us something we can use to help control future pandemics. If we can't stop them, The alternative is at least to allocate our resources better to try and reduce the number of cases in each place.
Looking back 100 years to the last pandemic also invites the question of 100 years from now, what will we be like in terms of how well we are protected against flu, or what things we could do to protect ourselves in some situations? Surely we'll be in a situation where we know more about what viruses are in the world and where they are. Surveillance will be so much better. Surely we'll be in a better situation where we have better drugs and better vaccines because we know more about the virus, so we know more about its weaknesses. We know more about our immune systems, and we know how to enable them to fight those pathogens better. At some point, we will have a universal vaccine that will protect us against flu for the rest of our lives. When this is a whole other question. There are a great number of approaches and really good ideas to doing this. There are already attempts at this that are at a quite advanced stage. It might be 10 years from now. It might be 20 or 30 or 40 years from now. We really don't know.
Crazy, huh? When was this made? So I put this on here. Published 11/30/2018. Is that bananas? So, this, I mean, people were talking about it that were in the field all day, every day. But, scientists knew that it was coming. We knew that there's going to be something at some point. We just didn't know when. And then, I loved how he said, like, oh, we'll be more prepared for the spread. We'll be more prepared how to control it. We'll be more prepared to be able to identify. And he says the influenza virus, it wasn't the influenza virus. It was the coronavirus. So we had the wrong virus. But, it was just, we didn't know. Nobody knows. So, I just think it's super interesting. The first patient was 1 year later, approximately. I just think it's a total trip. So we're going to talk about viral control today, but first let's finish off with our genetics talk. So we're going to talk about zoonotic diseases, and this is going to come up again and again when we talk about viruses. So zoonotic diseases are any disease that can be passed from animal to human, human to animal. That's what the definition of zoonotic means. So the problem with zoonotic diseases are these two things. We call them antigenic drift and antigenic shift. So drift versus shift. And so why do we worry about these two things, and what are scientists doing to talk about that and to try and figure out what's going on? So there's a little video on here if that will help you, but I'm just going to give you a quick little summary. So antigenic drift, this is, if you think of like drifting, is like aimlessly drifting. So what you would be doing is, like, on the virus, for example, like if these are the amino acids that make up the spike proteins of influenza virus, for example. So did you hear how he said H5, H1, H7? Remember, that was hyaluronidase. Remember, in the spike proteins of an enveloped virus, there are these spike proteins. And in influenza virus, which will be one of our cards, so it'll help you remember it also with one of our cards, there's H and N. So those spike proteins, there's a type that's called H and a type that's called N. So they call it H1 because it has a certain string of amino acids. They call it H2 because it has a different string of amino acids. H3 has a different string. So what's going on with drift, if we talk about the strings of amino acids that will make up this spike protein, let's just talk about, we're pretending it's a hyaluronidase string of amino acids. Then remember we talked about point mutations. And that point mutation, it can be a silent mutation. It's a good time to review. A frame shift mutation. What were our two others? nonsense and missense mutations. So there's 4 types of point mutations. Got all kinds of goodies today. There's chocolate down in there too if you were grabbing chocolate. Okay, so if there's one mutation in one of these amino acids in one of the viruses over long periods of time, that's called drift. So if there's a mutation, and then, for example, if this disease, if this influenza virus went from a pig, and some pig farmer, forgive me for my drawing, so went from a pig into a person, then the influenza virus, our bodies would probably recognize it as the influenza. It'd be like, oh, you know, I've seen that somewhere before. It looks familiar. Maybe there's an amino acid difference, maybe there's a couple amino acid difference. Then let's talk about shift. So it's been mutating in the pig for a long time, and these amino acids are all different. It's been mutating in the pig over and over. And then it gets into the human body, and the human body is like, I have never seen that before. I don't know what that is. So now the human body can't recognize it, and our immune system can't recognize it rapidly enough for it not to completely take over our body, for it to cause the cytopathic effects that it does. Yeah?
So for drift, there might be like a couple of changes, but our body can recognize it, but for shift, there's so many changes that we can't.
That our bodies don't recognize what it is anymore, and our bodies, the immune response is lower because our bodies don't recognize what it is yet. Yeah, we haven't really talked about the immune system yet, but we will. Yeah.
Does that mean that our bodies fight it off less or
Less.
fight it in?
Yeah, so
Okay.
our bodies, because we need to have time to mount an attack and recognize it as foreign. So by the time, you know, so for example, like my daughter gets the flu and I get the flu and we both have the same flu and we're both sick. If my husband gets the flu a couple months later, then I'm probably still immune to the flu, even though it might have a couple of amino acids different, because I just had it and my body recognized it. And even though it's mutated and there's small mutations, I probably won't get it again because my body already recognizes it. But if my husband's in the military and he comes home 5 years later with this flu and he's gotten this weird flu, it might take me down because my body doesn't recognize it, and so my body has to mount a response to it, and by that time it can be really effective, like as a virus, you know what I mean?
So the recognition happens after it's already entered the cells?
So
But we're speaking about the recognition in terms of the white blood cell response
White blood
after
cells.
it's in, rather than the cells, whether they let it in or not.
yeah, no, it's not whether the cells will let it in or not, it's whether the white blood cells will recognize it or not. Yes, great question. Yeah.
So it's more not whether the body fights it off, but the speed at which it fights it off. Like
Yes.
the immune system fights off a drift a lot quicker because they already know it.
Yes. And what the problem with influenza a lot of times is age. You know, our immune systems are slower, or youth, that they don't have the white blood cell army yet.
Mm-hmm.
So as we get older, our white blood cell army goes down. As we're younger, our white blood cell army is not mounted yet. And so our white blood cells are not able to recognize and fight off the virus before the virus wreaks havoc on our cells. Yeah.
Does antigenic drift and shift refer specifically to zoonotic diseases, or does it apply to all diseases?
We use those terms even for, we use it even in population genetics. So,
Okay.
yeah, even when you're talking about this population of deer went through antigenic shift, and then they can't mate with these group of deer anymore. So we use it as like a the amount of genetic changes. But that's what we worry about in zoonotic diseases, is the amount of genetic changes. that because that is, our body is not able to recognize it anymore when it goes through a lot of genetic changes.
Wait, when you're talking about the deer example though, that would be drift and shift of the genes of the deer. And we're talking about the shift and drift of the genes of the virus.
Yes,
Okay.
yes. Does that make sense? Does everybody
Yeah.
kind of understand that? Do you want me to go over that one more time?
No, that makes
So what I was saying was, we use those terms in like population genetics. So you have these deer that live over here. Okay, you have these deer that live over here, and there's a valley over here, and there's deer over here. And all of a sudden there's a flood and there's all this water and the deer can't get across anymore. So the genetic changes over here happen so much so that they can't breed together anymore. So we use that also in population genetics, but we worry about that a lot in viruses. Yeah.
Do viruses use the same amino acids that we use to make the protein?
Yeah, it's the same. Those are the only known amino acids. Yes, same amino acids. Great question. Yeah.
So are the mutations for the spikes happening within our cells then?
The
Or is it doing it on
mutations.
its, in a way like on its own, or does it need to be?
Yeah, it would be within our cells because that's the only way that they're replicating during the replication process. Yes.
They don't have the polymerase 1 to check it, right? That's why they
Yes. So,
mutate so fast.
yes, for RNA viruses specifically.
RNA.
Yeah, yes.
specifically.
And DNA viruses do have a high mutation rate as well, but yeah, I mean, RNA viruses, we would just assume that they're worse. And it's just kind of, we broadly throw that out there.
And the DNA for a spike protein comes from its own RNA then?
Comes from its own RNA. Yes, awesome. Great questions, man, you guys are awesome. I love all the questions so much because it really does help kind of guide. It really does help me a lot. I had one student in my last A&P class that asked so many questions, and you know, it's hard because you're trying to get through the lecture, but at the same time, like, there's such good questions. I feel like it helps everybody to feel like they can ask questions and, you know, brings up super interesting stuff where we can all kind of talk through it. I think it really helps. It's worth the time. Awesome. Okay. So antigenic shift versus antigenic drift, you can watch that video. So what's happening is there's just an accumulation of mutations versus big jumps in mutations that will happen in the animal that then that can be passed to humans.
And
Okay, so here are the zoonotic diseases that I just got this off the internet. So, and this, actually I got this off the internet my first semester where monkeypox was a big thing. Now, you know, we have all kinds of other things that we've been talking about, Zika viruses, and they still are talking about monkeypox. There's a new one that's going around. I don't know, I haven't kept up on my virology, but so here H1N1, H5N1, here's the bird flu. So we think SARS-CoV-2 came from bats and maybe pangolins. We weren't sure where it came from, but that's the going theory is where it came from a meat market. Hantavirus, rabies, these are all viruses. Chikungunya virus is from mosquitoes. So a lot of viruses that come from animals that we deal closely with around the world, and now because of worldwide travel, they spread like wildfire throughout populations. So it's also a good time to review endemic, epidemic, pandemic, review those terms again. Okay, so let's go through a life cycle of a virus. So I'm not gonna ask you step 1 through step 6. The reason I wanted to go through this is because it says it on your book. But the reason I don't wanna spend time talking about it is because I think it's pretty self-explanatory. Once I explain it once, you'll be like, oh, that makes sense. So how a virus gets into a cell is through, it's called adsorption. So what it does is the virus will attach to the human cell. So the human cell will recognize the virus, and the virus is really tricky. Those spike proteins or whatever capsid proteins or whatever's on the virus is very tricky, and it tells the cell to endocytose it. So it'll tell the cell, let me in, and the cell goes, oh, this is food. The cell is like, recognizes it incorrectly, is what generally happens. So now it recognizes it incorrectly, and then it'll start to endocytose the virus. So now it's going to, oh, I keep doing that. So it depends on the type of virus, but what happens sometimes for enveloped viruses is this envelope, because it's made up of phospholipids, just like our phospholipids, that it will mesh with the cytoplasmic membrane and just dump its contents out into the cell. So sometimes that will happen where the membrane just will adhere to our membrane. And I don't want you to memorize this either, just know the virus gets in. That's all it does. But let me show you just a couple ways that it gets in, just for your own peace of mind. So also what can happen is our cells can form a vacuole, like form a bubble around it, just like as if it were food or proteins or something that our cell needs. And then here's the virus inside. And then what happens is our cells have these things called lysozymes. And these are enzymes that will break up food that the cell eats. So the cell's gonna eat some food, and now these two fuse, and so that's how our cells digest stuff, is with lysozymes. So these two membranes will fuse, just like as if this were food, and break up this virus. Now, once it's broken up this virus, and now the cell thinks it's food, all this food, actually viral pieces plus the genome, is going to be released into the inner part of the cell. Now our own cells will replicate the genome, make the DNA and RNA or whatever it is, replicate the genome, or we'll use the enzymes from the virus. And then now it's going to assemble new virions. So let's just pretend this is a DNA strand. This is a DNA virus. Here's the nucleus of our epithelial cell. This DNA strand will get inside the nucleus. And now DNA polymerase is going to copy that viral genome, just like our own genome. And so when it copies our own genome, then it's making so many copies, so many copies, so many copies, so many copies, and it can lyse this nucleus. Now it's got a bunch of DNA and RNA, so now it's made RNA. Now it has DNA and RNA. Now what's gonna happen is the proteins are gonna assemble inside of our cells. Now they assemble, and now you have a whole entire cell full of virions, because our own cells replicated, our own cells made the RNA, transcribed, our own cells translated. So all of the virion will come together in a nice little neat package, and then I'll tell you what happens from here. So there's a couple ways it can damage the cell from here on out. So from the view of the virus, so like Dr. Harris was saying, from the view of the virus, you know, really, let's talk about like Staph aureus, for example. So Staph aureus is a bacteria. We're not talking about viruses for a second. It's a bacteria. This bacteria wants to eat, wants to replicate, wants to go on and live its life. But it doesn't want to do anything, right? Because it doesn't have a brain. It doesn't want things. It just does it. So they live in the most optimal environment because of their makeup, how they live. They live in a salty environment because that's their makeup. They live in different environments in the stomach because that's what the food is. So they're just living the dream, right? So if we're kind of like, it's called anthropomorphizing. If we're giving this virus a personality and giving it wants and needs, then it doesn't really want to kill everything around it, because then it doesn't have food and it can't replicate. Like, where is the cells so that it can replicate? It needs things to be able to survive and to replicate, right? So the worst kind of viruses or the worst kind of bacteria kill their host, because then it's like, well, But accidentally, sometimes they do. Sometimes they do kill their host. But how is this virus going to reproduce, going to perpetuate, going to make, live the dream, going to make its life the easiest? Well, what it can do is now, because there's so many virions, those virions can just burst this cell open and go out and infect new cells. So that's called lysing. So we already talked about that a little bit. So lysing a cell just means breaking a cell. Now those virions are gonna go all over and infect this cell next to it, infect this cell next to it, infect this cell next to it. It would only be limited by the amount of cells that are near it, or if it can get there, because they don't travel, they don't have a flagella. So it would just be what is near it. And now, to be able to infect cells before the immune system comes and takes over and recognizes it and kills it. So one thing it can do is overgrow and lyse the cell. So there's too many virions, it breaks out, and it can overgrow and lyse the cell. Another thing it can do to kill the cell, remember we're talking about cytopathic effects. Cyto means cell, path means like disease, pathology. So it's cytopathic effects. So it's also, it can take a portion of the cytoplasmic membrane with it. So say you have a DNA here from a virus, and you have these spike proteins that are floating around because the genome has been replicated and the proteins have been transcribed and translated. So it's just floating around in here. So what these spike proteins do is they come to the surface of our phospholipid bilayer and they arrange on our surface of the phospholipid bilayer like that. Now what happens is it starts to form a bubble. So now it starts to form a bubble like this, and now you have spike proteins. And now the genome will come inside to this virion, and it will take a portion of this cytoplasmic membrane with it. So now once it leaves a hole, and now this cell will die because it lost a portion of its cytoplasmic membrane. Depending on the virus, sometimes they can allow phospholipids to close the gap. It depends on the virus. Yeah?
Are they transcribed in the cytoplasm?
Yes, transcribed and translated in the cytoplasm, yes.
Okay, so this transcription happens in the nucleus for us?
Oh no, it is. No, you're right. It is in the nucleus. They're transcribed in the nucleus, and then but it depends actually, that's not true either, because if it has a replicase, it is in the cytoplasm. If it does not have its replicase, yeah, because a replicase can work in the cytoplasm to copy RNA to RNA, but to copy DNA to RNA and use our own RNA polymerase, it has to get into the nucleus.
Can't get into the nucleus? Or can't do
It
that?
can get into the nucleus, yes, yes. Yeah, great question. Okay, so it can take a portion of the cytoplasmic membrane with it. Another way it can kill our cells is syncytia. So what syncytia is when the virus is coming out and making new virions, and it's lysing the cell open. So now this virus will infect this cell and lyse this cell open. And now this virus will infect this cell and lyse this cell open. But what happens is they can form this big cellular glob, and those cells will all stick together and form a big glob around the viruses. And so around the virions. So what they're doing is they're forming a big cellular mess, kind of like tuberculosis does in the lungs with our white blood cells. But what they do is they'll form a big mess of cells around there so that they're hiding from the immune system.
Is it surrounded by a bilayer, by a cell membrane?
I don't think the cell membrane actually forms around it, but it's like they've come to the middle of like the phospholipid protein mess that they've made. I'm not 100% sure what the outer membrane and how the outer membrane is protected from the white blood cells, but basically it's like a big globular mess of all the cells, and they can go into the inside so that they protect themselves from the immune system. So what a syncytia is like a multinucleated blob of viruses and cell damage. And so like respiratory syncytial virus, RSV, forms syncytia. And so in the throat, in the respiratory cells, there'll be globs of messed up cells and the viruses on the inside. I'm sorry, hold on just one second. Yeah, go ahead.
Do you want us to know the methods of killing and hiding?
Yes, all the cytopathic effects. Yeah, great. Yeah, go ahead.
Oh, I'm sorry, I missed it. What causes the clogging?
So it's what happens. Let me show you. There's a video of it actually. It's like a 2-second video. Where is it? Okay. It doesn't even have any volume, but what it does is like it's the cytoplasmic membranes are dissolved or lysed.
Are each of these hexagons a cell?
Yeah, these are all like human cells. So now all the phospholipids are dissolved into like units of phospholipids, and the proteins are dissolved into units of proteins. And then because of this big mess on the outside, that the viruses can hide and reproduce on the inside and still be using all of our enzymes, because our enzymes are just floating in the ether.
And that cloud is like at base lysed cytoplasm?
Lysed cytoplasm, yes, yeah. So that's what syncytia are. So that's SARS can do that. So coronavirus can do that. HIV can do that. RSV can do that. Herpes. I don't want you to know examples. You don't have to memorize examples. Just memorize that that is a way that viruses can kill our cells. Okay, another way is that they can alter our own DNA or RNA or proteins. So they can alter our own DNA. They can alter our own RNA. They can alter our own proteins. And now our cell is malfunctioning. And so because that cell malfunctions, now we have the issues that result from that. So one of those issues that we'll talk about today is HIV. So HIV can also alter the genome of white blood cells, but so what happens is they're not working as well, or they're dying because it alters the DNA to where it doesn't function anymore. Yeah?
So each one of these 4 things is a different method of killing?
Yes,
Okay.
a different method of killing. So when it alters the DNA or RNA or proteins, probably not proteins, more DNA and RNA of a cell, that can lead to cancer. So these are called oncoviruses. So don't forget that term. Everybody make sure you know that term. An oncovirus is a virus that causes cancer. So can you guys think of one? Do I have any up there, for example? No. Can you guys think of a virus that causes cancer? It's okay if you can't, because we haven't really talked about very many of them yet. So like HPV, for example, causes cervical cancer. So we worry a lot about HPV, especially in youth, because some people can go decades and not even know that they ever had it. And then they have cervical cancer as they get older, and it's because the human papillomavirus affected the genome of those cervical cells, and because it affected the genome of those cervical cells, then the genome wasn't working correctly, the cells weren't reproducing correctly, and then all of a sudden you have cervical cancer, or your patient has cervical cancer, hopefully not. So we'll talk about more oncoviruses, more viruses that can lead to cancer. I'll show you a picture. So also what they can do is they can hide. So viruses can hide inside of cells really well, but also they can hide without necessarily killing the cell. But if they're hiding inside cells, we worry about that because they can reproduce anytime they want. Sometimes they'll hide and not reproduce, and then sometimes they'll just spring up and reproduce anytime they want. So for example, like herpes virus, like I get cold sores. So herpes virus, it will hide in the neurons of cells, I'll show you a picture of it, forever. And so it could just hide there, and then all of a sudden it'll start reproducing again, and then people can get an infection from that. Yeah.
Is that what shingles is?
Shingles, yes, that's from varicella zoster virus. It's the chickenpox.
So is it like hiding?
It's
Like
hiding,
this?
yes. So people that have the chickenpox have it forever, have varicella zoster forever.
Why isn't there a certain age group for when someone can get shingles after chickenpox?
Yeah, so the reason is because our immune systems get low as we get older, you know, so just it'll trigger the virus, that the virus can, sometimes it's like, the immune system is low, and so your patient gets something else, and that will trigger viral reactivation, or, you know, whatever it is that is affecting your patient that causes the virus to start replicating again. And because that virus starts replicating again, it'll reinfect the patient.
Is that the same reason why you just get a cold sore randomly?
Yeah, so it's like, they call them like fever blisters. So it can be from getting too much sun. It can be from getting, having a lower immune system. It can be from not getting enough sleep. Too much coffee is probably my
All of those
combination,
things sound
something
like,
that.
I
I'm
said,
sorry, what?
all of those things sound like code for lower immune system.
Yeah, it is all lower immune system, but sometimes they can be triggered by heat too, by temperature. So sometimes it can be like sun exposure, but it can be internal heat exposure as well, like fever.
Well, if you're out in the sun too long and you're not feeling well, isn't that a lowered immune system?
Yes, but also it's because of cellular damage, like because of the UV light, because of damage to the cells.
Okay.
Yeah, that's as far as I know really about herpes virus. I don't know a ton about it, but okay. So these are the two cycles that a provirus can go through: is the lytic cycle and the lysogenic cycle. So what a lytic cycle is when the virus will replicate and overgrow the cell, and a lysogenic cycle is when the viral genome is incorporated into the host genome. So if the viral genome is incorporated into the host genome, it's also called a provirus. It's called a provirus, but it will go through this lytic or lysogenic cycle. So a provirus is a virus that incorporates its genome into our genome. So make sure you know that. So we know shift and drift now, we kind of got that. Make sure to ask me if you want me to go over that again. And then lytic means when there's too many virions inside the cell and it will burst open. And this is a cycle. So this is when we're talking about like life cycles of the virus. We wouldn't really call them life cycles, we just call them cycles of the virus. But otherwise, it will incorporate into the genome of the host. So if you think of like lyso, it's lysing the genes, lysogenic cycle. So then it's incorporated into the genome of the host. And then every single time that cell replicates, it's also replicating the viral genome, and that viral genome is part of the human host forever, or pig, or cow, or sheep, or bird, or whatever it is. Okay, so here's what provirus is. When they incorporate into the genome, they can do that in two ways. So remember, this is a provirus. Is a virus that incorporates into the genome. So it can either do this, or what herpes virus does. Oh, well, we're talking about bacteria. I should not draw it that way. I'm sorry, let me fix this, because we're talking about a human genome. Here's the nucleus. We're talking about the human genome. These are the human chromosomes, are linear. Here are human chromosomes. Bacterial chromosomes are circular. Human chromosomes are straight. So these are human chromosomes on a human skin cell, like a respiratory cell or something like that. Depending on the type of cell, it could be totally different shape. But what can happen is the virus can incorporate somewhere into the genome, or what it can do is tether to the side of the genome. So what the DNA polymerase will do is just go all the way around and replicate the genome just like it's there. So it can tether to the genome, or it can incorporate into the genome. So that's what herpes simplex 1. Herpes simplex 1 is usually what causes cold sores, and that's they hide inside of neurons. This virus is tethered to the genome inside of neurons. I think like 80% of the population has herpes simplex 1. I can't remember exactly how many. Some estimates said maybe 100%, but who knows. So this herpes simplex 1, but not all people get cold sores because they have a low viral load, or they have, maybe it's in a different neuron or something like that. So these neurons can, if it goes into the lytic phase, from the lysogenic phase, it goes into the lytic phase, then it can overgrow and lyse and cause an infection. Yeah, go ahead, Doctor.
So can you, is it obvious, like if you were to look at the human genome after some virus incorporates its genome into yours, would it be obvious like what section of the DNA was incorporated, or you not be able to tell?
So usually what you can tell is they'll do DNA sequencing, but usually it'll attach to the same spot, to the same.
If you sequence the genome before and after, you'd be able to tell which part was inserted.
Yes, but usually what they do, because that's really hard to sequence the genome, and it's hard to know, sequencing the human genome is incredibly difficult. So what they'll usually do is test your antibodies in your immune system to see if they have any that recognize the virus. So we're gonna do that. That's exactly what happens when you take a COVID test. On the COVID test, we'll learn all about this, so don't stress about it right now. But there's little antibodies on the COVID strip, and your sputum runs through this litmus paper, so it diffuses by, and it will recognize if your antibodies will match the antibodies on the plate. So actually I should draw them like this. So these are your antibodies to coronavirus will be recognized by the plate. So
I was just wondering, maybe the technology isn't there yet, but maybe in the future could you use genetic engineering to remove that part? Because you said it's permanent once it gets into the genome, right? So, or is that just not a feasible thing to do?
Yeah, I mean, you would have to, how would you get the genome of that cell specifically without killing the cell? Am I saying
Well,
right?
yeah, I don't know.
Yeah,
Maybe
I don't know.
Maybe it's like a future thing, but
yeah.
Or if it's not even feasible, I think to identify it and then remove it the same way you can. Well, I don't know.
What would be cool, and if any of you are interested in it or have learned about it, there's something called CRISPR, and so CRISPR-Cas9, what it can do is recognize certain like AATTCCC, right. Whatever it is, that sequence, it'll recognize that sequence and cut it and remove it.
So that's kind of where I was going with it.
Yeah.
We learned a lot about CRISPR and biochem, and I figured maybe the technology's not there yet, but in the future could you identify regions that need to be removed and then remove them, and then you solve these issues with diseases or viruses that can't be treated, like HIV or something else.
Yeah, yes, they are 100% people are working on that for sure. Yeah, and they're a little bit worried about it because it's only A's, T's, G's, and C's. If it recognizes anything else, your patient could be deadly. So they're worried about it, but they're trying to perfect it. So yeah, I mean it's a great area.
For patients that are kind of already doomed.
Yeah, right.
What else could they be released?
Yeah, right, for sure. Yeah.
What causes something to go from the lysogenic cycle into the lactic cycle?
So we talked about that really quickly, but we can go over it. So sometimes it will just hide out there forever and never come out. and it will never enter the lytic cycle. But generally what happens is age or lower immune system or something will trigger, like if your patient gets a fever from something else or something else goes on that will trigger it to enter into the lytic cycle. Yeah. Cool. Okay, let's move on. Okay, so here's some examples of proviruses. HIV hides out in T cells, human white blood cells, T cells, and then herpes simplex virus hides out in nerve cells. Okay, so let's talk about oncoviruses. So human papillomavirus can cause cervical cancer. Epstein-Barr virus. Epstein-Barr virus is a type of a herpes virus that can cause lymphoma, can cause cancer in the lymph nodes. Nasopharyngeal cancer, it can cause stomach cancer. Hepatitis B, this is why we're crazy about vaccinating against hepatitis B, because it can cause liver cancer, and hepatitis C can also cause liver cancer. So those are the ones that we know of. Those are the most common anyway. So I know I'm not going to talk to you all about cancer, even though this is one of my favorite areas of research. But if you do have like 5 extra seconds in your life, and you probably don't, but if you do, then this is the reasons that cancer can initiate, the reasons that cancer begins. And so without going into all of these reasons, you know, in cancer bio, it's super interesting, you know, from a biochemical perspective of like what's going on with the biochemistry of the cell. You know, those Henrietta Lacks cells, those HeLa cells, how they can live forever, like how, and maybe that has something to do with our longevity and our immortality, you know, or maybe it's the way that we can figure out how to cure cancer. But I think it's super interesting, and so if you want to read through this on your own about how cancer is initiated and the things that trigger cancer, then you can read that through that. But in an oncovirus like Epstein-Barr virus or HPV, it's color-coded to the things that went wrong, that cell does to make your own cells damaged to cause the cancer itself. So you can just look at that. I won't ask you about it, but it's interesting. Yeah?
I was under the impression that all people have cancer cells. It's just that the difference between somebody who develops cancer and the rest of us is just that our immune system catches them and quickly enough and gets rid of them.
Yeah, so if any of our cells, we have broken cells always, you know, and we have cells that are damaged always. Like, you know, how many times have I been out in the sun and not put on sunscreen and have skin damage, you know? And so those are all DNA damage. And so, yeah, our immune system can, if our immune system catches it, most of the time, yes, for sure. And then sometimes our immune system can't catch it. And then sometimes the cancer cell itself has ways, now I'm anthropomorphizing cancer, but the cancer, the cell that has become cancerous starts out by evading the immune system. So depending on what the DNA damage is. Yeah. Awesome. Yeah.
Is all cancer that we know of the result of a cytopathic effect of an oncovirus?
No, so our cells, they will mutate on their own. So because of DNA polymerase 3, which we call it something else in humans, so I have been trying to not go back and forth with that, but we call DNA polymerase 3 something different in humans, which we'll talk about. But it has the same action as DNA polymerase 3. And then DNA polymerase 1 is the proofreader. So if the proofreader is not doing a good enough job, and that's usually what happens over a lifetime, DNA polymerase 1 stops working as well, DNA polymerase 3 starts making more mistakes, and so cancer is more frequent in older people. So yeah, that's from our own mutations. But yeah, viruses can, you know, for HPV, there's a high rate of cervical cancer from HPV, or hepatitis C is terrible also. Don't get hep C. Okay. So I already showed you the syncytia virus. So this shows you the absorption, the reproduction, how a virus can come in and out of cells, the viral life cycle. So you can watch that. That's super interesting. And then this one I just put on the end because I'm not going to go into it. because we're gonna run out of time, but you can look at it on your own if you're interested. This is called phage therapy. So this is like the new, well, it's not huge yet at this point, but they're using bacteriophages, so these are viruses that are called bacteriophages, to attack cancer cells or to attack other virally infected cells or things like that. So they're using phage therapy for all kinds of cool stuff now. Okay, so that was the end of viral genetics. So everybody stand up for a second, stretch, move around. We're gonna move into control and talk about vaccines. Oxygen, oh, I forgot our little teapot. Would you mind if I had you grab it? Yeah. It's, oh, I left it on top of the water station. Yeah. Oh, did you see it over there? Okay, so grab some tea if you need it. So vaccines, I think, are super cool to talk about because we've all had them, right? So what types of vaccines have we had? We might, hopefully we'll have time to talk about coronavirus, the new vaccine with coronavirus. Have you guys heard of that vaccine?
Yes.
Well, I know you've had it probably, but what type of virus or what type of vaccine it is? Do you guys remember from the whole news about crazy, all this new type of vaccine? Everybody was going bananas over it because it was a new style of vaccine. It was not FDA approved yet. Everybody was freaking out.
Is it like an RNA?
Yes, so it was an RNA vaccine. Yeah, so we'll talk, maybe you can, just spend 1 minute and, yeah, there you go. Spend 1 minute and talk to the people at your table. Everybody just take like 3 minutes. Talk to the people at your table or talk closely with the people and what is going on with an mRNA vaccine. So the vaccine is made up of RNA. So what's going on there when you're injected with RNA?
My guess
Maybe it takes that RNA and makes proteins that fights the virus. Like maybe it's coding for something that is right the virus.
Yeah, I don't really know anything about vaccine.
I don't know, I just got that.
That sounds pretty good though.
Well, I mean, if it'll do that with a viral RNA, could it do it with a vaccine RNA?
So what about regular vaccines, I guess?
Regular vaccines usually are the spike proteins that trigger your immune response.
Okay, yes. So an RNA vaccine. Maybe it's easier to make RNA. So like maybe we make the RNA and insert it ourselves. I'm not really sure.
Because if our immune response recognizes the spike proteins, then it will fight against COVID right away, or whatever the disease is. But the problem is the spike protein is mutates, right? So I don't know what the RNA is.
Okay. I mean, if they're moving to that now, it has to be like some sort of ease to make it, or easier to make.
Maybe cheaper to make, but also maybe more effective at variations.
Okay, let's go ahead and move on. So you guys can mull that one over and we'll talk about vaccines and what they are. So let's talk about viral prevention. So how do we prevent from getting a viral disease? So we're going to use the same physical and chemical control that we can use for bacteria. So that's cool. You know this. This looks familiar, right? We can call it a viricide or virustatic prevention. So are we going to keep the virus from replicating, or are we going to disassemble the virus or kill the virus or deactivate the virus? We wouldn't really call it kill it, right? Because it was not living in the first place. So we're going to inhibit viral activity or destroy the virus. So here are our same tools for physical or mechanical control. Here are our same tools for chemical control. So let's spend our time talking about vaccines. I don't know if anybody has one of these, but a lot of people in the UK have these and around the world. So some of my students from like Mexico or from parts of Asia have this scar. And what it is it's a smallpox vaccine. So we had a different smallpox vaccine in this country than they did in many other countries. So in the United States, if you were born in the United States, you likely don't have a scar that looks like this because of our vaccine was different. Crazy, right? Is that so crazy? Like when you go, if you travel, then you can look around on people's arms and see kind of like where they were born, because most people that were born in other countries have this scar that are approximately my age, maybe 10 years younger than me and older. So I'm 44, maybe 35 and older, all, but now in countries, most countries, they give the scar-free vaccine for smallpox. Okay, so in the video it talked about Edward Jenner. So this is one of our scientists that we're gonna know. So we are gonna know about Edward Jenner. It was in the 1800s, saved more lives than any other human. Everybody always says, right? On Wikipedia, you could flip through and every third scientist says that. But Alexander Fleming was a really cool guy that saved a lot of lives. And then this guy was the father of immunology that was the one who discovered how vaccines work. Never mind, it was completely unethical, but he did figure out how vaccines work, and so a lot of us are still alive because of him. So he was a zoologist and studied zoonotic diseases way back when. So,
From the video.
I'm sorry, what? This same guy from the video. Yeah. So this is Edward Jenner. So he was a wealthy guy and had a farm, and so his milkmaids were getting some sort of mild version of smallpox, but then they were all alive. And so he took a small boy that was under his employment and gave him cowpox. And so then he was like, let me give him smallpox and see if he'll live, which is terrifying, right? Like,
Can we call him Jenner the Jerk?
seriously, yes. Except he did save us all, so it's actually, this guy is our hero, this little kid. This little kid is our hero, actually. So this kid is the one who was the guinea pig, which is very illegal. And so he survived, and now we created vaccines. So what's happening, which I'm not gonna go into the immune system yet, you'll figure out all the immune system aspects, but what was happening is if you have two viruses, say we have two enveloped viruses, and here are the spike proteins. I know we only talk about enveloped viruses for some reason. So we have two viruses. One is smallpox, one is cowpox. And this cowpox has lower cytopathic effects than smallpox. This has higher cytopathic effects. But this, so what happens is your white blood cells, which we won't go into what white blood cells or anything like that. We'll figure that out later on in the semester. But this white blood cell is going to recognize this cowpox, and then it's going to be stored as a memory cell. And this memory cell that recognizes cowpox is stored forever. So when this same person gets smallpox, now because we've already made memory cells that will be triggered against the smallpox, then it will kill this smallpox before you even recognize that you're ill. So let me give you one example. Did I give you the example yet of my friend who's allergic to peanuts? No, I didn't give you that example yet. So he's a firefighter, he's a friend of my husband, and his daughter is allergic to peanuts on a deadly scale. So she was about 5, I think 4 or 5, and she went into full-blown anaphylaxis, throat swelling, can't breathe, blue around her lips, and her dad ran around the house and couldn't find the EpiPen. And it was within 30 seconds she stopped breathing. So he ran around, found the EpiPen, gave her the epinephrine, took her into the emergency department. They had figured out that the dog licked her, and they had given the dog food that had peanut butter in it. So it was within 30 seconds her throat closed. And that was because of her own immune system. So her own immune system is so strong and so powerful that in 30 seconds it found and attacked peanuts that it thought was foreign, that it is foreign to her body. That's not to most of ours, don't recognize peanuts as foreign. So, like for example, if I got COVID and then somebody else was next to me that already had it, they would still get the virus inside them. They're not protected on the outside from the virus. The virus still gets in there, but within 30 seconds, their white blood cells are attacking that virus so much so that you don't even recognize that you had it. So your white blood cells are that powerful. So that's what happened is with the cowpox, they triggered those white blood cells. So within 30 seconds of that smallpox inoculation, then that patient is completely immune to it. So that's what Edward Jenner did. So the last outbreak was in the 1950s. The WHO officially eradicated smallpox from the earth in 1980s. There are still some in laboratories in the United States and in Russia. Okay. So vaccines trigger our immune system. And so they trigger our immune system with things called antigens. So an antigen is just, the word antigen came from antibody generator, but an antigen is just anything foreign in the body. So it could be a spike protein of a virus, it could be lipopolysaccharides from a protein or from a bacteria, it could be peanuts for somebody that's allergic to peanuts, it could be grass for somebody that's allergic to grass, whatever it is that our body recognizes it as foreign, we call that an antigen. Okay, so here are the vaccines that we're going to learn about. We're going to learn about live attenuated vaccines, inactivated vaccines, conjugated vaccines. We won't have time to talk about mRNA or viral vector toxoids, but those are less used. So what an attenuated means is weak. So write that down, make sure you know that. An attenuated virus, it's live attenuated. So it means the virus is still living, which Dr. Harris and I, we're gonna stand on opposite sides of the rooms on that one. But live, nobody thinks they're alive, but it just means it's still active, but it's weakened. And the way that it's active and it's weakened is because of something called serial passage. So what serial passage is the virus in the lab, you use some sort of human cell type, and you'll pass that virus on to cell type to generation to generation to generation to generation. And it won't be exposed to any other viruses or any other cell types or any other anything. So what it's doing is almost like incest. Like when kings and queens all mated with each other from the same families, their genetics are weakened because they all have too similar of genome. So their genome is so similar, they're not going through mutations, they are not going through antigenic shift or antigenic drift for 10 plus years. So now this virus is not very good at replicating, it's not very good at transcribing or translating, it's not very good at getting into and out of cells. But what it is good at is it's injected into the human will recognize it, the white blood cells will recognize it as foreign, mount an attack on that virus and the new viruses that are good at replicating, that can propagate rapidly. So that's what a live attenuated virus is. So many of you probably have had MMR, that's the measles, mumps, rubella virus, rotavirus, smallpox, chickenpox, yellow fever are all live attenuated. Yeah?
Do you want us to know examples?
No. You don't have to know any examples, just know what the term means. Live attenuated means it's a weakened, active virus. Yeah?
So we weaken the virus
Yes.
by course for 10 years or whatever in a lab or
In a
something,
lab,
and then
yes.
taking the weakened ones.
Mhm.
Then how, when we inject that into a person, how does like the presentation of those weakened cells allow the body to fight off the non-weakened ones that would be in the environment?
Because the proteins are still the same, they're just the enzymes that it comes with, and maybe its proteins aren't as good at getting in and out because it's an older virus. So it's like using like the smallest kid on the little league team to come and fight the battle. You know, it still recognizes the virus, but the virus is not as functional.
But then so the body's used to fighting off the one from the lab attenuated vaccine.
Mm-hmm.
Then a new one that is functional comes into the environment. The cells are now, because they fought the one that they could eat, they now know the exact technique to use or something against
So
it.
it'll recognize it. So it's almost just like the cowpox example.
Yeah,
It'll recognize it
Just
still.
swallow it in.
Yes,
Okay.
it'll recognize it. Yeah, the white blood cells will recognize it. Awesome. So inactivated vaccines. So this is a vaccine that usually they use formaldehyde. So usually they use formaldehyde to break apart the lipids or to inactivate the enzymes, but basically they're including the whole virus, but the virus does not work. So that's an inactivated virus. Why do we use different types of vaccines? Because some are more effective than others, and we just try different stuff. Like maybe this one, like the influenza virus, the polio virus, rabies virus, Hep A virus, maybe it's not as effective. The most effective type of virus is a live attenuated virus. But what would be the problem with that?
You could get people sick.
Yeah, because it can still get people sick. It can still replicate. It can still incorporate into the genome or whatever does.
Didn't that happen with the polio vaccines?
Yeah, so there was a whole slew of polio vaccines that happened in Africa and in where was it? In Afghanistan. And so a lot of people, I was gonna bring this up at some point, but it's a good point, is that some people are very anti-vaccine in those parts of the world. And so a lot of people from Western cultures are judgmental, like, oh, it's not voodoo, like get your vaccines. But actually there was a whole generation of children that have polio from getting a polio vaccine. And there was all kinds of problems with it in the 70s and 80s and 90s. So, you know, it's just something to think about, of being respectful of other people's thoughts around it, because it is scary. You know, you're injecting your child with the virus. And so, you know, we're all trying to protect our children, and we all want our children to not get ill, but it's scary also. These need refrigeration, so that's something to think about in, you know, parts of the world that don't have refrigeration. So inactivated vaccines, so we talked about that, formaldehyde. So a subunit or a conjugate vaccine, so what a conjugate vaccine is just, they'll take one piece. So they'll take this whole virus and they'll just take one little protein of this virus and inject that protein into the person. So now it'll trigger the white blood cells, and now hopefully when the virus comes, that it'll recognize that virus as foreign and kill it. But it's not as effective, and sometimes it needs what's called an adjuvant. So it needs to hook this protein to this other large protein, because it might be too small to trigger a big enough response from your white blood cells. So that's a conjugate vaccine, is a small piece. Conjugate, small piece. mRNA vaccines, you guys can talk through those together. I think that's really cool. Recombinant, let's skip those two because we're gonna do this in the lab. And then toxoid vaccines. These are bacterial examples that create exotoxins, so we can have a vaccine against the exotoxins. That's just something to think about. So here are our vaccines that are viral vaccines: hepatitis B, rotavirus, which is an intestinal virus, poliovirus, COVID-19, influenza, MMR, varicella, which is chickenpox or shingles, Hep A, hepatitis A, which is foodborne, Human papillomavirus is a sexually transmitted virus. And meningococcal, that's bacterial, we didn't need to talk about that. And then identification, so remember prevention, identification, treatment. Identification, we're gonna do this in the lab, so I'm not gonna stress about it now, but it's called serotesting. That's what you do with the COVID strip. You do what's called serotesting, so we'll show you what that means. But then let's talk about treatment. Let's talk about antivirals. So everybody really quickly tell me what you think the problem is with antivirals.
It tastes terrible.
Antivirals taste terrible?
The COVID ones too.
Oh, oh, I've never tried those. Oh, crazy. Yeah, so that is a problem. Yes, antiviral resistance is a huge problem because of the mutation rates. Very good point. What is a problem when you're coming up with a pharmaceutical that will target a virus? Yes, very good point. It's very hard to do because they're obligate intracellular. So how are you going to get the pharmaceutical into the cell without killing the cell? Very good point. And then there's one more point. So that you have a pharmaceutical, you're on a pharmaceutical team that's creating a drug that's going to attack a virus. How is that drug going to attack a virus? I know I'm asking it in a kind of a broad way, so I'm not. Yeah.
It's going to be attacking your own cells as well.
Yeah, because well, depending on what it is. So why? You guys think of why? Like what? So what does beta lactams? What do they target? They target the bacterial transpeptidase enzyme. So what, as an antiviral, what do you make? What is the problem? So I know I'm asking it in a weird way, so I'm trying to feed it to you, but without giving you too much information. But so you have capsids, and in some viruses, you have that. You have capsids and a genome. What's your target? Maybe the capsids? Well, they're a protein, and they're not really that much different than our proteins. Well, maybe the replicase enzyme, if that comes with one, maybe the reverse transcriptase enzyme if it comes with one, but that's kind of all of our targets. What are we gonna target? You know, we don't have all of the ribosomes, name all the ones from the antibiotic targets. Ribosomes, cell wall, DNA replication, because of that weird topoisomerase. And what was the other one? Folate, the metabolic synthesis that we don't have. So they're all targets of things that we don't have. So yes, they're intracellular, very hard to target. Yes, their mutation rate is super high, very hard to find something that will not change. That is probably our biggest problem. But also, we are out of targets. We don't have very many ways for antivirals to kill the virus. So we're only gonna remember one suffix. That's it. We're not gonna remember any of these. We're not gonna remember all of these, but we're just gonna remember one suffix. So if you see this VIR or IVIR, that is an antiviral. So something that says VIR at the end is an antiviral. So that's all we have to remember. VIR, antiviral. So like acyclovir, valcyclovir, people were taking for a long time Relenza. What is that? What they were taking for coronavirus, for COVID? I forget. But so these are antivirals. They're, physicians are very hesitant to prescribe antivirals because of viral resistance, and sometimes they don't work that well. I take acyclovir if I'm feeling like I'm getting a cold sore, and it makes it go away in like 3 days versus 9 days. So it does help sometimes. So here are the targets. So here's neuraminidase inhibitors. Sorry, you can watch that here. Neuraminidase inhibitors, RNA replication inhibitors. So those for replicase, DNA replication inhibitors. So For herpes virus, I don't know if that has a reverse transcriptase, but if it has a DNA replication inhibitor, and then HIV is a reverse transcriptase inhibitor, but the reason, oh, here I have a slide on this. So here we started making antivirals, but we haven't made a lot of antivirals because they're not very effective. And HIV antivirals are so effective now because they're making a cocktail of different drugs and different amounts of different pharmaceuticals in one prescription that will prevent viral replication, and they have to change it constantly. So it's like $30,000 a month or something like that, ridiculous for HIV antivirals, but now they're giving up. I think the price has gone down recently, but scary. So there's so much trickier to target because those three things we just talked about, natural antivirals exist. So it's not very many natural antivirals exist. Remember, we didn't have very many synthetic antibiotics. Most of our antibiotics were non-synthetic. They came from other things. They came from penicillium mold. They came from other bacteria. So we don't really know how to invent things from scratch. We're not very good at that. So we are trying to make things from scratch, but there's not a lot of natural antivirals that exist. Yeah?
Is there any way you could like target them based on size?
Based on size?
Since viruses are a lot smaller, right? So
not that I know of, because they're also the same size as, like, you know, proteins and other things.
right.
So like
maybe something that like kills the RNA or DNA inside of it, but something that can only get into small proteins.
Yeah, I'm not sure. I don't know kind of the new targets that they're doing, but yeah, I think it's cool. It's a really cool area of research. So resistance is a huge problem in antivirals because are they using our DNA polymerase 1 or not? Are they able to proofread or not? Okay, so here are our viral diseases. I'll go through these in the review session. Varicella zoster, that's chickenpox, influenza, rabies, rubella, rotavirus. So this is our virus McHappy. That's my mnemonic, because there was a viral name that fit under all of those. It's harder to come up with a mnemonic than you think, so sorry it's so dumb. So virus McHappy is the way I did a mnemonic. And then here are the viral diseases that fall in under those branches. So here's our viral wrap-ups. You guys can read through those on your own. Here's some term review from when we talked about bacteria. We talk about transmission in the same way. So DDX, we're not going to get time because we're going over time, but I did want to make sure, you know, I'll go through this on, I'm going to do a review video tomorrow, so I'll talk a little bit about if it's viral or bacterial and how to tell the difference between those. Yeah?
Do we need to understand the RNA vaccines?
No, we'll talk about that as we get more towards the final. Yeah, so okay. RNA vaccine, hopefully you guys talked through it, but it's an RNA code that codes for spike proteins. So yeah, I won't ask you about it just because we didn't have time to talk about it, but you're injecting the person with RNA, and that RNA is able to get into the cell, and our own ribosomes will create spike proteins. And those spike proteins, now our cells will recognize those as foreign. So we're making spike proteins ourselves. Kind of crazy.
Okay, so it's not the complete RNA of the virus, just the spike protein part.
Yes.
Okay.
Awesome. So we'll talk about, so here's our bacterial and viral infections that are similar. Respiratory infections, a lot of us will get a respiratory infection. Is it viral? Is it bacterial? We don't know. That's probably the most questionable. Is it viral or is it bacterial infection if you have a cold or cough? So here's the way to tell-ish. Don't ever take this to the bank. Always ask your physician, but just kind of a broad way to be able to tell. Here's otitis media, that's an ear infection. A little bit of a way to tell maybe if it's viral or bacterial. Viral or bacterial conjunctivitis, so an infection of the eyes, that's called pink eye. Here's gastroenteritis, so also it is hard to tell gastroenteritis. Is it viral or is it bacterial? Usually if it's viral or bacterial, you kind of know where you got it. Did I get food poisoning? If you got food poisoning, it's probably bacterial. If you, I don't know, maybe it could be viral as well. So those are really hard to tell gastrointestinal infections. Viral meningitis, if it's meningitis, you always treat it as if it's bacterial first because it's deadly. So you treat it as if it's bacterial, then they'll test the cerebrospinal fluid and know whether it's bacterial or viral. So they'll do a spinal tap, take out the cerebrospinal fluid to test if it's a meningeal infection of the meninges to see if it's bacterial or viral.
What does CSF mean?
Cerebrospinal fluid.
Okay.
Oh yeah, great question. So I'll talk about this on the video. Okay, so we're not gonna have time to talk about prions. We'll talk about that next time. Let's do our note card, and then yeah, we're gonna do our note card, and then I'll play my recording for you in the lab. So this is human immunodeficiency virus. We don't need to say that, right? Everybody knows. One thing I do really want you to know, so on your coronavirus card or somewhere in your notes, make sure you know the difference between what coronavirus, what that means when somebody's saying that, what it means when somebody says SARS-CoV-2, and what it means when somebody says COVID-19. What do those terms mean? So I think that's really important to know, so make sure you know that, and I can go through that with you if you need to. And then, yeah, we can talk about it in the lab if you want to as well. We have kind of a short lab today, and then I'm going to play a little audio recording for you.
For the midterm, for the note card specifically, can you post somewhere, like, just to make sure? Cause I'm not sure if I'm missing any.
Oh yes, for sure.
They're
Yep,
kind of
I'll post them all today.
Okay,
Yes.
thank you.
Yeah, I don't want to forget to do that. Yeah.
How are the note cards presented in questions?
So note cards I'll make them a little bit easier for the midterm, so that you can just identify what is Gardnerella vaginalis. What is, so just doing some little identification. So not to worry about spelling, not to worry about coming up with the bacteria or virus yourself from a question, but matching. So like, okay, what is sexually transmitted? What is not sexually transmitted? What is a probiotic? What is not a probiotic? What is gastrointestinal? What's a skin infection? So being able to identify some of those like main things about each one. So like, you know, C. diff is a super infection. It causes endospores. Some things that are like identifiers of those note cards. And then when we get towards the final, we're gonna do diagnostics. So we're gonna be able to see like my patient has this. I'm gonna pull from my note card knowledge and be able to identify what that might be. Okay, human immunodeficiency virus or HIV. It's a positive RNA virus, single-stranded RNA virus. It's enveloped. It comes with its own reverse transcriptase. Now you know what this means. It is a provirus. So its genome is incorporated to our own genome. It is not an oncovirus. There's a few different types, HIV-1, HIV-2. You don't have to know any of those. I just thought that they're interesting.
Do we need to know that it's not an oncovirus?
Here, not an oncovirus. No, just know that it is a provirus. Yeah, you know HIV doesn't cause cancer. So here is the cell type that it infects. So this might be foreign to you right now, but it infects T cells. You could just say T cells, but you eventually will know what CD4 positive T cell is and what that means when we get into our immunology section. Patients will generally die from a secondary infection due to a low T cell count. And a patient will acquire AIDS, which is acquired immunodeficiency syndrome. And when it's considered AIDS, they have less than 200 T cells per cubic meter of blood. Then it's officially an AIDS diagnosis. Your patient might have flu-like symptoms that are chronic, weird rashes, night sweats, sore throat, swollen lymph nodes, long-term, secondary infections from a low white blood cell count. So this is the quintessential immunodeficient patient, because they are immunodeficient because they don't have an immune system, because they're missing part of their white blood cells. Arguably their most important white blood cell. So here, just as a little cartoon, if you want to look at what HIV looks like, here's HIV versus SARS-CoV-2, what those look like and the difference between those, if you want to look at that. How many people have HIV around the world? Yeah, so it's a big problem. Okay, so I will see you guys in the lab. So you can go ahead and get all of your PPE on, make sure you have all your PPE on, and then